Welcome back to part 2 of erythropoiesis, where I told in the previous video where I will elaborate in depth about the precursor cells. So precursor cells are nothing but the erythroblasts. Blast cells are the one that tends to this word blast. So the blast cells are the one that tends to lay down the lineage for the next upcoming steps of the erythropoiesis under the precursor cells. Now, before this normoblast or the erythroblast, which is nothing but the precursor cells are formed, they are inevitably formed first from the pronormoblast. That which you can see over there. So pronormoblast gets converted into normoblast. This normoblast is nothing but the precursor cells. Pronormoblast is the first cell in the erythroid series. So therefore, its size is going to be very huge. That is going to be around 20 to 25 micrometer. This is the one that you tend to identify first in the erythroid series. Now, cytoplasm of the pronormoblast is going to be very little. It is going to be occupying only 20% of the cell. I understand. I will show you the picture of how a pronormoblast looks after I describe about it. So, after that, you see something called as polyribosomes. Now, what are these polyribosomes? They are like small granule-like structures that you will find inside the cell. Okay, so that is what a polyribosome is. What happens if there is polyribosomes? Now, in the pronormoblast, the polyribosomes are very high in content. You have a lot of polyribosomes in the pronormoblast. Why? Why? Because they can make our study more complicated about the erythroid series. Not only that, this tends to make the cytoplasm intensely basophilic. Basophilic means blue. Now, what about the nucleus? The nucleus is going to take up 80% of the cell. It is going to be very huge, and the mitosis is present. Now, why should there be mitosis? So that it does not stop at pronormoblast. It goes ahead and continues into the early erythroblast or the early normoblast. Now, what about the hemoglobin content? No hemoglobin content yet. Now, how does it look like? Keep these things in your head. That is the largest cell, 20%, 80%, and the basophilic. This is how a proerythroblast looks like. All right? Fine. Now, we will go to the next topic. When the pronormoblast undergoes mitosis, it gets converted into the second precursor cell, that is the early normoblast. Obviously, the size is going to decrease because in my previous videos, I have already told you the erythropoiesis is a progression of a decreasing size. That is why your size of the pronormoblast is as high as 20 to 25 micrometer, but your RBC is just 7 to 8 micrometer. Okay? Your early normoblast, its size is going to decrease to as low as 15 to 18 micrometer. All right? Now, the cytoplasm of this is going to be deep blue basophilic as usual. Now, because it is basophilic, you can understand that there is a very good amount of polyribosome content that is also present. Okay? Now, because of this basophilic erythroblast, because of the presence of this basophilic cytoplasm due to polyribosomes, the early normoblast is also known as the dash. The hint and the clue is a cloud called as the fun fact. Comment down below as what could be the other name of the early normoblast. Now we'll talk about the nucleus of it. The nucleus is going to be made up of heterochromatin clumps. Also, it tends to appear like wheel spokes. Now, what about mitosis? Mitosis obviously has to be present so that it can go to the next step, that is the intermediate normoblast. The intermediate normoblast is nothing but the second year of MBBS. So the mitosis is obviously going to be present, and the first year is going to get converted into the intermediate normoblast. Then what is a pronormoblast? Pronormoblast is during the 11th and 12th, the NEAT preparation. Hemoglobin. The hemoglobin tends to appear for the first time. This could be a very important MCQ that you should know. The stage where the hemoglobin appears for the first time is the early normoblast. Now that you know this much, now how are you going to remember such an important MCQ? Now, if you've seen Frozen, majority of you must have seen Frozen, there is this song Elsa sings that is first time and forever. So she was early bird. She was just an early bird who was learning how to control her magic. So similarly, the early normoblast, the hemoglobin appears for the first time. Now, the intermediate normoblast, obviously the size is going to increase to a huge amount. If you stopped and if you're thinking, what did I just say? Did I say the size is going to increase? Well, that means you're paying very good attention to the video. You're absolutely correct. The size is going to decrease and not increase. So, what's its size going to be then? Its size is going to be 0 micrometer, is it? No. It's going to be 12 to 15 micrometer. Then what about its cytoplasm? The cytoplasm intermediate changes into something else. All right. So this is what the cytoplasm would show. And why does this happen? Why is there such a change happening? The hemoglobin that is present is going to increase in the intermediate normoblast, due to which it tends to dilute the PRP. PRB over here stands for polyribosomes. Therefore, since the polyribosome contributes to the basophilic, which is currently being diluted by the hemoglobin, which is a red colored substance, the cytoplasm changes from blue to pink. Then what about the nucleus? This gives the appearance of the checkerboard. I know I did not show you the picture of the early normoblast and the intermediate normoblast yet. I will show it to you after these two topics are done so that you'll be able to correlate better. Now, why do we say intermediate normoblast as polychromatic erythroblast? The other name for intermediate normoblast is polychromatic erythroblast. Presence of, hemoglobin is an acidic structure. So due to the presence of this acidic hemoglobin, along with the eosinophilic RNA, this makes the cytoplasm polychromatic in color. Now, this is the reason why we also tell intermediate normoblast is also known as the polychromatic normoblast. Okay. Now, you must have got the answer for this thing, that what is the reason for the eosinophilic nature of the cytoplasm? Yes, it is obviously due to the presence of RNA material. Okay. Now, mitosis, do you think it should be good, or do you think it should start becoming slightly sluggish? Intermediate is already done. The second year is done. The third year is called as the honeymoon period. So the mitosis is going to become slightly sluggish. I'm going to take a deep break from whatever my work is to do. This is how the basophilic erythroblast and the polychromatic erythroblast looks like. The basophilic erythroblast, I know I have already told you the answer to what early normoblast's other name is, but yes, you can just look at it. So the early normoblast getting converted into the intermediate normoblast, this is how they are going to look like. Late normoblast. This is going to be like the third year. We are in the process of converting from the second year to the third year, but we have not yet entered into the gist, or we yet do not start preparing for the PG. The size is obviously going to decrease to as small as 7 to 12 micrometer. What about the mitosis? Think about it. In the intermediate normoblast, the mitosis is almost given upon us. So the mitosis is going to be absent. Okay. Now, orthochromatic erythroblast. This late normoblast is also known as the orthochromatic erythroblast. Why? Why? Because the scientist said so. What do you mean by the word ortho? I mean to say ortho, same, chromatic, color. Same color as what? Same color as the upcoming RBC. The RBC appears red in color, right? So the orthochromatic, the cytoplasm of the orthochromatic starts to take up the slight pinkish to red color. That is why the late normoblast is also known as the orthochromatic erythroblast. The nucleus. Have you ever heard of an RBC having nucleus? Well, if you did, that is considered as an abnormal RBC. You should not be having a nucleus in your RBC. The nucleus is going to become very small and pyknotic. That you can see over here how small the nucleus has become. Now, the hemoglobin is going to increase to a huge extent over here. Now, juvenile. Mature cells are nothing but the reticulocytes, the ones that you can see over here. This is the reticulocyte. Now, before this reticulocyte is going to form, what's going to happen is this orthochromatic erythroblast has a small and a pyknotic nucleus. This small and pyknotic nucleus tends to disintegrate and gets kicked out of the cell, like you can see over here. While we are done with everything and we are in a very strict PG preparation from third year to fourth year, throughout the fourth year, the hemoglobin is going to keep increasing a lot. Size is going to be slightly larger than the RBC, but definitely smaller than the orthochromatic erythroblast or the late normoblast. Now, why do we call this reticular? Reticular because it appears like a mesh. Reticular is like a mesh like appearance or of something. A reticular network like appearance it has. Now, due to the reticular network of appearance this mature or the juvenile cell has, we call this as the reticulocyte. And after this, we're done with our fourth year, getting ready for our exams, getting ready for our graduation. I don't know if that looks like a graduation cap, but then yes, it is a graduation cap. So finally, we have become erythrocytes. Cleared our PG's size is between the range of 7 to 8 micrometer. The shape is biconcave. Proerythroblast gets converted into the basophilic erythroblast, gets converted into intermediate normoblast, also known as the polychromatic erythroblast. The polychromatic erythroblast gets converted into late normoblast. Late normoblast is nothing but the orthochromatic erythroblast. During this process, the small and the pyknotic nucleus tends to get kicked out of the cell. The reticulocyte is formed and the nucleus is separated, and therefore the RBC does not have any more of the nucleus. We're finally going to have the erythrocyte formation where we are finally done and graduated from our MBBS, and that is the end of the precursor cells. And I will meet you in the part 3 of erythropoiesis.